I'm very happy to start off to introduce Dr. Noelle Bowlin. She's going to be talking about the California Cooperative Oceanic Fisheries investigations. Probably most of you that know about this know of it as CalCOFI. Dr. Bowlin has a master's and a PhD from Scripps at the University of California. She's a fisheries research biologist at the Ecosystem Science Division at the NOAA Southwest Fisheries Science Center, and she's the lead of CalCOFI. And I had the pleasure over the last few years of working with Noelle through the North Pacific Marine Science Organization, or PICES, on some of the work that we've been collaborating across the country and really across the Pacific on small pelagics. Noelle is an ecosystem modeler, and she looks at the mesopelagic zone and climate variability within the California current and how that has impacts on pelagic species such as sardines and anchovies there. She has a particular focus on larval dynamics and looking at habitat use, environmental variability, and anthropogenic stressors. And so she'll be talking about the CalCOFI program. This is a really phenomenal program. It's one of the oldest fisheries monitoring programs in the country. It's 75 years old and came into fruition in the 1940s in response to the collapse of the Pacific Sardine fisheries. And it's really a pioneer in looking at ecosystem approaches to fishery management. So it integrates data and various understandings on predators, on competition, on prey sources, on ecosystem interactions, and it's constantly bringing in new technology in terms of how they operate. And so Noelle will talk a little bit about the history of the program, some of their current focus, and really we're trying to see what we can learn from their program and how that approach might have applications in Alaska. So thank you for coming, Noelle, and the floor is yours.
Hi, everybody. It's hard to put into words how excited I am to be here. First of all, I've never been to Alaska. What it looked like initially, the whole west coast there, from California down into Baja and to the Sea of Cortez. And then now what we have here on the right. And so the red box is what we do quarterly. We do quarterly surveys, and we've done this for a little over 40 years consistently now, quarterly surveys of the red box. We do winter, spring, summer, and fall. We have a cruise out right now. And the winter and spring cruises, they expand north. That's what's north of that red box. And in the blue, you also see the National Marine Sanctuaries there that we have also incorporated more stations recently in partnership with the sanctuaries. I don't have a lot of time to talk about that today, but I would love to talk with you if you're interested in it later. So anyways, we've changed a bit. But this is where we've landed, at least for right now, and we're still trying to change some more. I'll share that with you. And so what do our current sampling efforts look like? Well, we have a lot of things going on, as many of you do as well. We have visual observations, obviously the physical from the CTD and the bottle samples. We have our net tows. We have underway data. We do environmental data. There's a lot of things happening. And we don't have time to talk about all of it, obviously, but for a moment, I'm going to focus on the net tows because, again, like I told you, I'll take a portion of the time to talk about ichthyoplankton ecology, which is what my aspect of the program focuses on. And so in the beginning, there was the baby sardine. And I have to tell you that when I started, I was a technician in CalCOFI. I've spent my entire career in CalCOFI. And I was an ichthyoplankton sorter, like Dracus was as well. So good to see you. And I didn't know what fish larvae looked like. I studied terrestrial ecology in college. And when I first saw this baby, I was like, oh my gosh, that is the cutest thing. I was very excited. You have to be inspired by what you study. You have to be, right? And it's whatever gets you. And I remember thinking, wow, that little thing, it's so small. That's about 5 millimeters long. And I looked outside at the ocean and I thought, how do they make it? And I love that that is my job. I get to try to figure out how these fishes make it and how we can make it so they get to continue making it. But back to the beginning. So they knew what sardines look like. But in those plankton samples, which are in the nets, in those plankton samples were not just sardines. And at the time, they got all kinds of things. By the way, these are just some of my favorites. Obviously, many of these are mesopelagics because I love the mesopelagic, but we get, as you probably all know, most fishes in the ocean have a pelagic larval duration, and so they're all in the plankton. And so regardless of where they settle out as adults, we are catching so many of them in the plankton. So a lot of people ask, well, but you're talking about fisheries, and we catch adults and consume adults and utilize adults, but you're studying larvae. Well, that's because we can catch them. Adults are, as you know, very good at swimming away from nets, and it takes a lot more resources to get those adults. Anyways, so why? And these two right here, Albert Ahlstrom, the first director of CalCOFI, and the Southwest Fisheries Science Center, affectionately called Ollie, and Jeff Mosier. These two were at the beginning, and they said, well, we're collecting these plankton jars, and our charge is to figure out the ontogeny of sardines. However, in those jars, there's so much else in there. There's so many other things, and sardines don't live in a vacuum. They live in a dynamic environment, interacting potentially with all kinds of other species. And so what Ollie and Jeff said was, well, if we are going to be collecting all of these organisms, we need to figure out what they are, and we need to make that quantitative, because who knows what kinds of questions will be asked, and maybe they can be answered with some of the things that we find out. And so they went to finding out. Now, this wasn't like the main thrust of CalCOFI, but Ollie was able to convince the feds that he should be supported to figure out what was in those jars. And so he did with his team. And there are two people that remain from his original team that are my current teammates. And together, over the course of 40 years of collection of samples, they published the ontogeny and systematic of fishes, as well as our CalCOFI Atlas 33, which is the early life history description of all the fishes in the California current, the early life history, I should say. And again, just a favorite example of a dragonfish and what it looks like as a baby. My son loves this one too, because it looks like an alien. Or he says, well, Mama, that could be a slug. Okay, fair, very fair. And so that was just a moment to talk about that. But in terms of the larval fish abundance data, they're so incredibly useful. And so we use them for what CalCOFI was developed for, which is a fisheries focused program. Originally, that's what it was focused on. So for fishery management plans and stock assessments for both, the two columns on the left are federally managed species, and the three columns on the right are state managed species. And these are just some examples of the ones in California that CalCOFI data are actively used to help in management. And I mean, the data are used for all kinds of things, and it was hard for me to pick out what to share with you that could maybe touch some of what you do and help spark some ideas for how we could work together, because we very much want to connect with you and move right on up the coast. That's our plan. But I know you have an IEA, and we also have an IEA, an integrated ecosystem assessment. And so the CalCOFI data are utilized in the ecosystem status report, which is presented to the Pacific Fishery Management Council, which is upcoming in March. The data are also used in the condition reports for the national marine sanctuaries, and this is to assess the resources in the sanctuaries. So our data are being utilized, which is the goal: make the data available and make them usable and user-friendly. That's the idea. And so if we think about how some of the data are being used with Ollie and Jeff's idea of make sure we do it right, make sure it lasts, and it's quantitative so that people later can come along with new technologies to answer unresolved questions. And so our colleagues were able to utilize the archive of ichthyoplankton samples and look at the anchovies when anchovies were booming, and then they just seemed to disappear in the mid-90s. Well, using compound-specific stable isotopes of Rasmus, our colleague, Scripps CalCOFI is the group he's with, in the leg of that stool, the Scripps leg, and other colleagues to look at trophic length of the prey that the anchovy larvae were consuming, and when they moved higher on the food chain, where they were consuming higher food chain length prey, they didn't do so well. They seemed to just disappear, which is very interesting. Lower on the lower food chain length meant boom, which is wild. We just didn't know those things, and we didn't have the ability to answer that question with the tools that were available in the 90s when they went away. And then again, just trying to pick quick examples. Another example of that, another colleague, Erica Mason, she is now a stock assessment scientist in the Antarctic program in the Ecosystem Science Division at our lab. And Erica was able to use machine learning along with morphological IDs. So several of us in our larval lab, we took the old archive samples of paralabrax species, our sea basses, which are high dollar items. There's 3 species. And for sport rec fishing in California, they are a big deal. But they're really hard to tell apart, especially in the early stages. And so Erica was able to utilize, we all got together to figure out some pigment patterns, and then she implemented some machine learning with those pigment patterns. And now that information, her model, is being used, well, it's under review by the state to be included in their management plan, which is pretty exciting. I know I've talked to some fishers, and they're excited about it too. Zach Gold, who is in the audience here today, Zach rolled in with as a PhD student, he said, I want to get in on this eDNA thing. I'm super interested in it. And at the time, that was brand new when Zach was a PhD student and then moved into a postdoc with us and is now at PMEL running some omics shows, which is cool. Check out his poster. It's really good. Anyways, Zach was able to reconstruct what the composition of our samples look like with eDNA, this idea that you can take a sample and run the genetics in a sample without destroying the sample so that those samples can be left for other researchers to come and ask questions and utilize them for answers later, that just blew our minds. It's very exciting because for any of you that collect any data, and the lengths that we all go through to do it systematically, to archive it and to curate it and to make it available, it's hard to impress upon people that don't do that type of work. And I know that you've all touched that in your own careers. But people that don't, it's hard. What I'm saying is people that don't, I mean funding sources. It's hard to impress upon folks how important that work is so that people like Zach, people like Erica Mason, people like Rasmus Swalethorpe can come and do this work to answer important questions that we all need to better manage our fisheries. Back to talking about sampling. So we zoomed in for a moment to take a look at some examples, and let me zoom out. So these are our current sampling efforts. And what does that really mean, just doing this all? We went from a fisheries-focused program in the beginning to a marine ecosystem monitoring program, doing a lot of different things, as many of us do. We do this, right? And so the reason I tell you, not because you don't know, is because there are a lot of data. They're available. We want you to use them. We want you to take a look at them. We want you to say, oh, hey, no, I have this idea. You want to work on this thing? Or, hey, actually, I've been doing this thing that I think you guys could do better. I took a look at your data. Or something like that. Whatever it is, let's do it. Let's talk about it. It's so cool that we get to work on these types of things. What I want you to take from this image is not all the different data streams that we've had started, stopped, started in the middle, or, because, again, we're not perfect. The idea is that we're trying to get an idea of what's going on in the ocean and getting all the parameters that we can to write these stories, to answer the questions, so that all of the senators, I am blown away by Senator Sullivan's super engagement here. I think it's super cool, both Murkowski, and so that was really interesting to have legislators that know what's going on so closely with what you're doing. I want everybody to have ideas of what we're all doing to try to describe the ecosystem that we all need to sustainably exploit. And so that's why I put this here, just to know that we have data. We'd love it if you'd use it. And we'd also love it if you have ideas for us on how to do it better. And so I started with telling you that we have this collaboration, but collaboration is key. And I give you this example where we got together with these other programs up and down the coast to say, hey, we're monitoring, you're monitoring. That's great. Let's put our data together and make it so that the ocean observing network across the world understands how important fisheries surveys are. Because what I learned from a colleague was that, because I've only ever been in fisheries for my career, and she had always been in ocean observing, she's like, people don't really know much about the fisheries survey work that you guys all do in the ocean observing space. And I was like, oh, really? I didn't know that. Because in ocean observing, in the OOSes, if you will, a lot of the projects there are funded on cycles on 1 to 3 years. Fisheries surveys are not. We have mandates we have to meet, and so we have to monitor all the time. And so the whole point of this was, hey, OOSes, we're here. Let's work together. And also to bring our programs together up and down the coast, to say we cannot stay in our silos, because that is a dangerous place to be, especially as we're trying to understand all of the changes that are happening with our climate, the change in administration. You just never know whether that's state, that's federal, whatever it is. We need to stick together. We need to be aware of what we're all doing. Let's not reinvent wheels and waste time. Let's do it together. And that was the point of this project that we did. And so I'm bringing you to now, into the future. And a couple of things. My original plan a few weeks ago was to end right here, but I won't end here. First, let me talk about this, what we're doing. So our center, the Southwest Fisheries Science Center, reorganized, and we created a new division, the Ecosystem Science Division. CalCOFI moved out of our Fisheries Resources Division into the Ecosystem one, so my program moved. And with that was the Antarctic program. We're together because we do very similar work, CalCOFI in the Antarctic, just in different places. And then also we have an offshore wind program. And in that program, we don't know what's going to happen there, but we do know that offshore development of any kind is going to be on the table. It's just going to be on the table. And the idea here is, what we're doing right now is there are 2 areas off of California that have already been leased to offshore wind development companies. And this one is off Morro Bay, and here's one off Humboldt. And so we have a new system, it's kind of like a library or an army, whatever you want to call it, of underwater gliders. And what we're trying to do is, right now, do 2 things with the gliders. We're getting out there to see what type of data we can collect with those gliders, because both of these areas, particularly this one off Morro Bay, are within the CalCOFI grid. And so we have 75 years of data within which to put the glider data in terms of context. Because at some point, if and when these wind turbines go into the water, we're going to lose access to our traditional survey methods. Putting big white NOAA ships out there, big UNOS vessels, we're likely not going to be able to do that. And so while we have time, we're trying to do both things at once. That's one effort. And then another piece of that is we can deploy gliders, as you all know, as you probably do. We can deploy gliders much more quickly than we can deploy a big white ship. And so that's another piece that's happening. And when we want to deploy quickly, that would be in response to something, if needed, as I'll show you in the slide that I'll end on in a moment. And so just in November, we had the first glider survey off of Humboldt here. This one was done. Again, and the reason why there's a line here is it's because it's 2 companies that have this area, these leases here. And so we're collecting acoustics, basic physical parameters, as well as shadow graph images to look at plankton. And then right now, like I told you, we have the NOAA ship Reuben Lasker. See, Reuben Lasker is out right now, and for the winter CalCOFI. And so what we were able to do is utilize a different method for adding stations to our CalCOFI grid that's here. We have a fixed grid, basic oceanographic survey, right? We have a fixed grid. But for this, what we did is we divided this into substrata and employed a probabilistic method in terms of choosing stations, such that it can be a little bit more random. You know, we could definitely talk about spatial autocorrelation later, but this sets us up so that when we have additional time, depending on how much time we have, we can run that and depending on what the ship is, grab some additional stations, which we're doing right now. Again, we don't know for how much longer we're going to be able to collect this information with ships in these areas, but we're also we're going to do it for as long as we can, while also putting in the gliders to get that information. And so, you know, let me just say this in case one of you wants to ask the question, well, what if you are told that there won't be wind energy development going on? Well, what we do know is that offshore development of anything, a deployment of anything, maybe it's a data center, you know, that's put underwater because it's cooler, will be put in place. Or maybe some other energy source will be collected. It's going to require assessment and monitoring, and we would still employ these same methods for doing that. And so if it's wind, if it's something else, we're ready to go. That's what we're trying to do. And so these are the places that CalCOFI is heading in the future, and I hope that we can do this together. We're trying to, like I said, learn and employ new techniques to answer unresolved questions. The one that's always going to hang out there is recruitment. That's a tough one, and obviously it's species specific, but I think we've made some headway, certainly with anchovy. We're learning a little bit more about sardine, only after 75 years, but we're working on it. And then another piece that's become just so apparent to me is when there are perturbations in the system, and when I mean system, I mean the California current, where CalCOFI samples, I will say that we are looked upon, that our program is asked nearly right away every single time, hey, what do you guys have on this? We have this situation. I don't know how many of you have heard of the rediscovery of the DDT barrels that were dumped legally off of Los Angeles in the 40s, 50s, and 60s, but predominantly by Montrose, or at least the production was by them. But they were legally dumped back then. They have been rediscovered, and in the sense that there was a sanctioned dumping ground, and then what David Valentine from UC Santa Barbara found in a different survey, he was like, what is that? They were doing ROV work and acoustic work, and the barrels are outside of the legal dumping area. Very far afield, they haven't found the end of that field yet after multiple surveys. And so we've been asked, well, what does CalCOFI have? Both state and the feds asked us that. And well, we have samples from that time. And I told you, I do mesopelagics. Well, I used to. Hopefully get back to it soon. But what's special about mesopelagics is that a lot of them, half of them, are vertical migrators. And so they are moving up and down in the water column, and we've got samples from that time, from that place, before the dumping and during the dumping, and now after the dumping. So again, it's when there's perturbations in the system, all of us, when we have time series data, when we have these monitoring data, are so critical that we do everything we can to protect these time series, because they provide context. They answer the questions. What is this change? What is this change from just a simple perturbation, whether it's anthropogenic or, well, tricky to say climate, but you know what I mean. How do we do that? Here's another example. This one hits really close to home for me. I'm from Los Angeles originally. I grew up there. My aunt and uncle lost their house 3 weeks ago, and it touches us. It's really hard, and we're left feeling very helpless. And our cruise was out at that time, and they left January 3rd from San Diego on the Reuben Lasker, and just 3 days after the fires were roaring, They moved right in here to Santa Monica Bay to do their sampling. And I'm getting phone calls constantly, Noelle, it's snowing ash. And all of that, everybody was it was very much, it's snowing ash from the fire. These are people's homes falling on us. And so what we were able to do, because we have to, we sampled as much as we could in the area per our scheduled samples. And then when the ship got a little bit farther north, we were able to bring the ship in. We sent new gear to do other types of sampling with our colleagues at Scripps, UCLA, and NC State, a lot of places, some fire scientists that we talked with for trace metals, for aerosols, that types of thing. And then we redirected the NOAA ship back down to the area to resample the area. And I show this picture here. Here are two of my teammates. This is our surface net.
So I just have one final question, and then we'll let you go. I was wondering if you could elaborate a little bit more about the inverse relationship you saw between food chain length and I believe it was sardine recruitment, and if you saw that in other ecosystem dynamics, if that relationship is held over time.
Yeah, thanks for the question, Matt. And it was for anchovy because that's who was around then, because after the sardines went away, the anchovies arrived. There's a whole story behind that too, which is quite fun, and we can chat about it later. But it looks like, so if you notice, the samples that were analyzed went through from the 90s, from the 90s through 2005. And so that didn't include present time because we also ran out of anchovies. But that's the only species that we've done that work on. But over that whole period, that relationship held up. And it was pretty interesting. And it looked like what became very apparent is that we had to look at a specific early life stage. It wasn't all larvae, because by the time those larvae were, if you will, post-flexion, they had, their swimming fins and such, not all their pigment, their diet shifts again. This is what we're learning. We just learned about their diet through looking at their muscle tissue and the isotopes. And yes, it was conserved. It was only for that species over that time period when we had samples, when anchovy were in the system. And so we had them in the system again now. We are seeing the largest abundances of anchovy consistently since 2014 that we've seen in the entire CalCOFI record. Which is wild. It's so many, and that's all the way through for us for CalCOFI, and then the other programs at our center that collect adults. It's the same. So we're starting to look at that again, to look